All right. Probably going to split this chapter into two because there's a lot of material in here. And it's an important chapter. And most of it is prokaryotic, but there's a little bit of eukaryotic, which frankly you should already know because you've already had general biology, and many of you have already had A&P. And a ribosome is a ribosome is a ribosome wherever you find it in. Well, a few differences, but anyway. Okay, so let me introduce this picture to you. This is a type of microscopy called dark field microscopy. And you may have already guessed that you're looking at protozoa, but there's something interesting here, and that is the guys that you think are protozoa are this guy and this guy and these two guys right over here. These are paramecium. You've seen paramecium before, you know, big guy like that, and it's got cilia all around him. And when you're looking at them on a microscope, they are humongous. They're really big. So, I mean, you have no problem finding them on 10x at all. Well, protozoa are eukaryotic. Obviously, they have a nucleus, even though you can't see it from this picture. But look at this big boy right here. This is called Epilopecium, and he is a bacterium. So I wanted to point out you can't go by size on eukaryotic versus prokaryotic. The reason he is a bacterium, prokaryotic, is he has no nucleus. He's way bigger than any eukaryotic cell that you see there, though. So that then takes us to the first thing in this chapter, the difference between prokaryotic and eukaryotic cells. All right, so things that are important for life. This is always covered in the first chapter of biology. You've covered it before. All things grow and get bigger. All things reproduce on their own, independent of anything else. One cell, for example, splits into two cells. It doesn't have to be told to do it, or doesn't have to interact with another cell to undergo mitosis. All cells are responsive. For example, some cells move towards light, like some photosynthetic organisms do. Bacteria, for example, if you put a drop of sugar solution on a slide, they will move towards the sugar solution. That's responsiveness. All cells, all things that are alive, metabolize. There are different metabolic cycles, obviously, but they all have to make energy. Now, there's one guy that doesn't fit here. I've already talked about it in Chapter 1. Viruses do not fit. They do not adhere to these rules of life. They don't grow on their own. They don't reproduce on their own. They don't metabolize. They use the host cell's metabolism for the production of new viruses. They use host cell machinery for reproduction. They have the blueprint, but the host cell makes them. They don't respond. They're more like chemical structures, chemical molecules, than they are live microorganisms. So anyway, read up on that in this chapter. Okay, so I've already delineated the major difference between prokaryotic and eukaryotic cells, but here it is laid out even more. Look at the examples given at the bottom of each bulleted list. The picture over here, again, just to make you think about your assumptions. Your eukaryotic cell is this guy right here, and a prokaryotic cell is this guy right here. It's not that much smaller than the eukaryotic. The eukaryotic guy on the right is a photosynthetic algae, single-celled algae. He's got some flagella that you see right here in the front, and there's a nucleus right here, and there's some chloroplasts that you see in green. This is a electron micrograph, color enhanced, by the way. And, you know, there's lysosomes and blah, 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 mitochondria, but they're kind of hard to delineate in here. Now, look at the prokaryotic guy on the left, and you don't notice a heck of a lot. These little salt and pepper-like things you see here inside are ribosomes, but you see no ER, and you don't see any chloroplasts. Really, all you see is some cell wall, which is that kind of a dark gray. And then this thing in pink is a capsule, although we haven't talked about it. And there's just not a heck of a lot inside the cytoplasm. Again, this is a picture out of your textbook delineating prokaryotic and eukaryotic. Electron micrograph next to the diagram. Okay, so we're going to spend most of our time on prokaryotic cells here. You're looking at electron micrograph on the left showing two bacteria that have divided. It may look to you, for example, like this is a nucleus right here. It is not. It's actually DNA. That is true, but the DNA is not enclosed in a nuclear membrane. And then here's a look at the bacterium growing on an agar plate. Okay, so we're going to start external and go inward. All right, so bacterial cells. Okay, let me draw a bacterial cell here. The cell wall is in blue, and let me maybe make a cell membrane in red inside of the wall. And let's do in yellow. Okay, so all bacteria, well, 99.999%, have this cell membrane and a cell wall on the outside. And then some bacteria have on the outside a coat. Let's see, let me change the setup here. Okay, make a coat that I'm doing in yellow right now. It's on the outside of the cell wall, and it's called a glycocalyx. There we go. And it can be thick, like I drew it. In fact, it could even be thicker than that. You just saw one in pink on that previous picture. I pointed it out. It was alongside the eukaryotic cell next to it. This is called a capsule, and it's a polysaccharide usually, but it can be a polypeptide. And if I erase my capsule out here, alright, and I go back and make, let's see, make it thin. Alright, so now I'm drawing another one, but notice that this is much thinner than the other yellow thing. This is a slime layer. Slime layers are thinner. They're less organized. Slime layers are for attachment, like attachment to teeth and things like that. Used in biofilms, by the way, for the attachment to surface structures. They're for protection. Capsules, being much thicker, have some added benefits. They really protect a lot because they have a thicker layer, so they protect against phagocytosis. So a white blood cell cannot phagocytose this cell effectively because it has that capsule. Capsules also keep the cell from desiccating, drying out. Also, capsules, because they are generally polysaccharide or slime layers, can be used actually as an energy source, kind of like eating your own coat if you had a food coat. Okay, fimbriae. If you look at the picture in the middle here, you see a couple of bacteria. Look at these red fibers. They may look to you like flagella, but they're not. They are more rigid. You'll see a better picture coming up. They are fimbriae. That's plural. One is called a fimbra, although bacteria have more than one. Therefore, adherence, attachment. Again, also, they'll come up in biofilms to attach to surfaces. They're made of protein. Also, some viruses. Bacteria have viruses, and viruses can attach to these fimbriae in order to enhance their infection of the bacterial cell. By the way, this is a capsule stain that you're looking at over here. The bacteria are the small dark purple cells that you see inside, but the capsules are the clear areas around them. They look like little halos around the cells because they have no charge. They don't pick up any dye. Okay, now, pilus. I'll show you a picture of a pilus coming up here in a minute. They're rather like fimbriae, except fimbriae are solid. There you're looking at a fimbriae on end. A pilus looks like a fimbriae, except it's hollow. I'm trying to draw a hole. It's not working very well. Well, let me do this. There. Okay. So a pilus is hollow. They are attachment structures between bacteria, but their job is for sexual reproduction. They are conduits for DNA being transferred from bacterium to bacterium. We'll talk way more about that later. We have a couple of chapters on genetics coming up. Again, plural, pili. Okay, flagella. You're probably used to flagella. Alright, so here's some different movements of flagella, of flagellated bacteria, right here. And the type of motility is determined somewhat by the number of flagella the bacterium has and the arrangement of the flagella. You see a flagella stain right there. The bacterium is the big, well, this thing right here. And notice that the flagella are actually bigger, longer than the cell. So here's the setup on the flagellum. Here is the cell membrane. Here is the cell wall. And notice that the flagellum has a basal structure that goes through the wall and the membrane. Then there's a kind of an angled hook-like structure. And then the flagellum is coming out that way. Now, look at the movement of flagella. They're different than eukaryotic flagella. Eukaryotic flagellum, if you're like a euglena or something, you have flagellum coming out of your body, out of your cell membrane, and it's used kind of like a whip, as if you took a belt and you just went back and forth with your hand with a belt in it. In the world of bacteria, the hook rotates around. You can see that circle right there. And as it does so, it causes the flagellum to rotate around kind of like a propeller on a boat. So it's a very different kind of movement than eukaryotic flagella. Okay, arrangements. I'm going to show you some examples in lecture and some motility videos, but let me just lay out the structures here. Okay, so at the top left at like 11 o'clock, you've got a single flagellum. By the way, these are, for the most part, except for one, electron micrographs. Monotrichous. Trick means hair. Mono, one. Then you've got, over on the right at about 2 o'clock, amphitrichous. You've got a flagellum coming out at each end. By the way, nice view of a spiral-shaped bacterium. So you can see a flagellum coming out both ends. Amphi, prefix like the word ambi, like ambidextrous. You can use both hands. Ambi, amphi, both ends. Okay, peritrichous. Look at the two examples at about 3 o'clock. You see flagella coming out all over the cell. Peri means around, like the perimeter of a room, the area round. Then at about 9 o'clock and 8 o'clock, you see a bunch of flagella coming out only at one end. That's called a tuft. It's called lophotrichous. Lopho means tuft. Now most bacteria, well, let me back up. Lots of bacteria don't have flagella, nor do they have movement. Staphylococcus, streptococcus, they don't have any motility structures, nor are they motile. But a lot of bacteria are motile by flagella, like E. coli, like Salmonella, like Bacillus. Now, there's a different kind of motility structure that some bacteria have. They're called axial filaments, sometimes also called endoflagella. So you're looking at a bacterial cell here at the bottom in lavender, and notice that there's kind of a blue-like structure wrapped around the cell. And you can see it maybe a little bit better down there on the electron micrograph. You can just make out that dark hair-like structure wrapped. And here you're looking at it in a cross-section. Here's the actual cell. I'll put a C right there. And then there's a wall structure out here, and you can see some little tiny things right in here. Those are the axial filaments, and they create a kind of an odd motility where they pull in, and it causes a corkscrew-like movement of the bacterium. And the important guide that, or there's a class of bacteria, I should say, that use that type of motility. Again, I'll show you examples in lecture. The spirochetes, of which syphilis is in. Okay. So, here's a look at the left-hand side. You can see flagella and fimbriae. And fimbriae are light red coming out. And over at about 5 o'clock at the bottom right, you see on the left bacterial cell fimbriae, and then you see a big pilus. This is called a conjugation pilus. It attaches the cells, and again, this is a conduit for DNA transfer. It's hollow. Okay, walls. This is really important. In fact, it's the most important thing we've covered, because this totally relates to staining in bacteria, which is going to become very important. Most bacteria, 99.99999%, have cell walls. And most of those bacteria that have cell walls have a very important, rather unique chemical structure called peptidoglycan. Remember, we're still talking about bacteria. There are actually 3 different components you see here that are in cell walls. Not all bacterial cell walls, but in some bacterial walls. Peptidoglycan, lipopolysaccharide, and teichoic acids. We'll talk about all 3. Now, let me focus on peptidoglycan, which should tell you peptides, protein-like, and amino acid-like, and glycan for glucose. Alright, so look at this thing over here. You're looking at 2 molecules bonded together, acetylglucosamine and acetylmuramic acid, and these 2 ringed compounds are variations of glucose. And they form these long chains. You can see one of these chains right here, and it's alternating molecules of acetylmuramic acid, NAM, with acetylglucosamine, NAG, with another muramic acid, with another glucosamine. Then you see some long strands over here on either side, and then you see some long strands of them below and strands above, and it forms like a lattice work structure. And this is what gives the strength of bacterial cell walls, is this PG. I'm going to call it from now on PG, peptidoglycan. By the way, on that same picture I was just using, there's some little, what look like blue pearls coming down from the muramic acid and linking to the lattice strings below of more muramic acid and glucosamine, and that holds all of these strands of molecules together. That's the peptido reference in the name. And again, you see another variation of the wall on a picture to the left. Okay, so that's peptidoglycan. It can be thick, it can be thin in the wall. Now, some bacteria have lipopolysaccharide, I'm going to call it LPS, lipid and polysaccharide, and some bacteria have teichoic acids. But again, PG is the most common thing in the walls. Okay, so the thickness of the walls, the amount of peptidoglycan, and whether a bacterial cell has teichoic acids or lipopolysaccharide, gives us 2 categories of bacteria. Bacteria are divided, eubacteria. Remember, eubacteria are true bacteria. I'm not talking about archaea. They have a different architecture. The bacteria are divided up into 2 groups. It's like humans are divided into men and women. Well, bacteria are divided into 2 groups by their cell wall. you see the two categories here side by side on a picture, gram-positive versus gram-negative. Look at the green layer first. The green layer is PG. By the way, the cell membrane is in black. Forget that. Don't confuse cell membrane with cell wall. Now, that's gram-positive on the left. Now look at the gram-negative on the right, and you see a very thin green PG layer. Gram-positives contain teichoic acids. You don't see them on the picture, but they do. And gram-negatives do not. Now, gram-negatives have something that gram-positives don't have. They also have peptidoglycan, but it's much thinner. But on the outside, you see this as this red layer. I don't know why they made it zigzaggy, because it's not. But outside here, there's this layer, which is the outer part of the wall, and it's called lipopolysaccharide. Gram-positives do not have that layer. By the way, lipopolysaccharides, also called endotoxin, because the major component of this LPS is called lipid A, and it's a toxin. We're going to talk much more about endotoxins later, but they're equal terms, endotoxin, lipopolysaccharide. Now, bacteria can have extra things in their walls. For example, we're going to talk, coming up quickly actually, about the acid-fast bacteria. I've already mentioned Mycobacterium tuberculosis causes TB. That's a group of bacteria called the acid-fast. They stain differently. And in addition to PG, and in addition to teichoic acids, they also have a waxy-like component, mycolic acid, that's rather unique for them. Trying to get the... There we go. Alright. Okay, so here's another look at the walls. What you're looking at here is a side-by-side gram stain. Gram-positive on the left, gram-negative on the right. You see that they stain differently. We'll get into why and what the stain is like and what the stains are in the next chapter. But if you look on the chart on the table here, you see thickness of wall, gram-positive, peptidoglycan content, gram-positive, teichoic acid, gram-positive. But then if you look over at the gram-negative, you see lipid content, way more protein, lipopolysaccharide, and you can see that there are some other differences like sensitivity to penicillin antibiotic or other chemicals. And on the picture, again, another way to look at the gram-positive and gram-negative walls. So up here, you're looking at gram-positive. The PG is in pink. PG, pink. Teichoic acid, these things embedded in there. On the picture down at the bottom, look at the PG right there. Thin pink. But here's that lipopolysaccharide layer right there that you do not see in the gram-positives. And again, remember, don't confuse a cell membrane, which is what you see at the very bottom, with a cell wall. Okay, so variations in cell walls. Archaea, a different group of bacteria, they have no PG. They have other things. They also have different cell membranes. They live in extreme harsh conditions. They have to have different components, different chemicals in their walls and membranes to be able to resist the high salt, high heat, high acidity, blah, blah, blah. Okay, we're not going to talk very much about them because they're not causes of disease in humans. All right, acid-fast bacteria. I've already mentioned it a couple of times, and here you're looking at a slide. The big blue cells are human cells, white blood cells and such, but look at these very thin, kind of a pinkish bacteria. That's your Mycobacterium bacteria. Again, acid fast because of the wax component to the other stuff in their wall that other bacteria don't have. This is an acid fast stain. We'll talk about it in the next chapter. Then we've got bacteria that don't even have a wall. I mentioned that 99.999% of bacteria do, but these guys don't. This is Mycoplasma. Do not confuse it with Mycobacteria. It never has a wall. And if you look at the picture, you'll notice that it has all sorts of weird shapes. Look at that guy right there, or this Y-shaped here. That's because cell wall confers rigidity, and without a wall, they have a membrane, but without a wall, there's no continuity in shape. The wall components, thickness, chemicals can affect all sorts of things, features of the cells. We'll talk more about that coming up in other chapters. Membrane. Not much to say about a membrane. You've studied this in A&P, you've studied in general biology. It's the phospholipid molecules. You see them in blue and yellow. So remember also that it's a bilayer. Okay, so 2 layers there. Here's the outside of the cell. Here's the inside of the cell. If you took one of these guys and blew it up, you'd have a phosphate head and 2 fatty acid tails right there. Okay. Then you've got these proteins embedded into the cell membrane. Not much else to say. You know that the function of a membrane is transport, cell transport, so they're selectively permeable. In bacteria, it's the same thing, except there's a couple of extra things that the membrane does for bacteria. Photosynthesis, if you're talking about a photosynthetic bacterium, oops, somehow I just lost everything. Well, let me get back to where I was. Oh, there we are. Bounce me out. There are a lot of photosynthetic bacteria, and of course bacteria also have to undergo cell respiration for energy production to get ATP. Your cells, other eukaryotic cells, have mitochondria for that, or they have chloroplasts for photosynthesis for this to occur in. Bacteria don't have those things. So their cell membrane actually, the inside of the cell membrane, this area, let me get rid of that, down in under here, is where cell respiration and photosynthesis occurs, because you have to have a membrane present for that to happen. Okay, I'm going to stop here, and we'll finish up this on video 2 for this chapter.